So as we know, glycolysis is a process in which the breakdowns of glucose occur in presence of certain enzyme to generate NADH and ATP. So two phases of the glycolysis is preparatory phase and payoff phase. So in this video we discuss about the preparatory phase. We already know what is preparatory phase in the earlier videos, so please check out the earlier video. So the first step of the preparatory phase is the conversion of glucose to glucose 6-phosphate in presence of hexokinase. This is the energy dependent step. ATP is needed, Mg2 plus is also needed. So this is the structure of glucose. Now glucose, so ATP is needed, ATP is transformed into ADP. So the gamma phosphate of ATP is transported to the 6 carbon of the glucose. That is why it is called glucose 6-phosphate. So the gamma phosphate of ATP is trans is transported to the 6 carbon of the glucose. That is why it is called glucose 6-phosphate. So glucose is transformed into glucose 6-phosphate in presence of ATP and Mg2 plus. This reaction is catalyzed by hexokinase. The del G value is minus 16.7 kg per mole. This is why it is a exergonic reaction and also spontaneous reaction. And the next step is the glucose 6-phosphate is transformed into fructose 6-phosphate in presence of an important enzyme that is called as phosphohexokinase, or you can say phosphoglucokinase. So this is a reversible step. Mg2 plus is also needed in this step. So the glucose 6-phosphate is transformed. This is the transforms fructose 6-phosphate. This is the structure of fructose 6-phosphate, and this is a reversible step of the
Sorry, there is a mistake.
glycolysis.
This is not
There is a mistake that
phosphohexokinase. This is phosphohexose isomerase or phosphoglucose isomerase, because the second step is the isomerization step. So the second, this is isomerase, not kinase. Now the third step of the glycolysis is very important. That is phosphorylation of fructose 6-phosphate to fructose 1,6 bisphosphate in presence of an enzyme phosphofructokinase 1. It is a regulatory step of the glycolysis, and we already discussed about the phosphofructokinase 1 in the earlier videos, so please check out the earlier video. So this is the structure of fructose 1,6 bisphosphate. As you can see, the 6 carbon is phosphorylated. Now ATP and Mg2 plus is needed in this step. This reaction catalyzed by phosphofructokinase 1. In case of plant and others, some organism, this is phosphofructokinase. So the gamma phosphate is transported to the 1 carbon and fructose 1,6 bisphosphate in form. Why it is called bisphosphate? Because as you can see, the two phosphate remain in two different carbon atom, 1 and 6. That is why it is called fructose 1,6 bisphosphate. Now fourth step of the glycolysis. So first there is a point. In case of plant and some bacteria and protist, the phosphofructokinase 1 is absent. In this case it is called as phosphofructokinase, and there is no ATP is needed. PPI is needed in this step. Mg2 plus is needed and fructose 1,6 bisphosphate is formed. So in this case phosphofructokinase transports the phosphate from PPI to the fructose 6-phosphate and form fructose 1,6 bisphosphate. The fourth step of the glycolysis is fructose 1,6 bisphosphate to dihydroxy acetone phosphate and glyceral dehyde 3-phosphate. This compound 3 carbon compound, and it is also a reversible step of the glycolysis. So in this step
This is the structure of fructose 1,6 bisphosphate. So this is the structure of fructose 1,6 bisphosphate, and this reaction is catalyzed by aldolase. So this reaction is catalyzed by aldolase. This is the structure of dihydroxy acetone phosphate. So this is dry hydroxy acetone phosphate, and this is a 3 carbon compound. On the other hand, another 3 carbon compound is formed, that is called as glyceral dehyde 3 phosphate. So glyceral dehyde 3 phosphate is also formed from the fructose 1,6 bisphosphate. So these are the three two compound is formed from fructose 1,6 bisphosphate. The daily value is 23.8 kJ. It is an intergonic reaction because the value of the free energy change is positive. Now, the fifth and last step of the preparatory phase of the glycolysis is dihydroxy acetone phosphate to glyceraldehyde 3-phosphate, because glyceraldehyde 3-phosphate enters into the payoff phase of the glycolysis. So, this is the structure of dihydroxy acetone phosphate. It is also a reversible step of the glycolysis, and glyceraldehyde reversible step of the glycolysis. This reaction is catalyzed by triose phosphate isomerase. So this is the structure of dihydroxy acetone phosphate. Dihydroxy acetone phosphate transform into glyceraldehyde 3-phosphate in presence of enzyme triose phosphate isomerase. Now this is an important thing. This is the structure of diglyceraldehyde. The structure of diglyceraldehyde 3-phosphate. So diglyceryl phosphate. Now the carbon atoms, so derived from the glucose. The first carbon, that is 4 or 3. The second carbon is derived from 5 or 2 of the glucose, and the third carbon, that is derived from 6